PhD. He was trained, started kind of jumping on the butterfly insect train when he was a postdoc at the University of College London with Jim Mallet. So he's been at Stride for about 10 years. And in addition to being a staff scientist, he's the Dean of Academic Programs there. So he organizes all the internships, graduate student visits, all that sort of thing for people interested in tropical biology. So it's really great to have him here at BYU, and we're hoping that we can facilitate opportunities like that for students who might be able to come and visit Panama. But I'll let him do the rest of the introductions, and let's welcome Owen.
All right, thank you, Paul. Can everyone hear me? So my voice is carrying perfect. Yeah, so before I begin, I mean, as Paul said, I wear two hats at Stride. I'm a staff scientist, and I'm going to talk about a lot of the work I do today over the next 50 or so minutes. But I'm also the academic dean, which means I'm responsible for the scholarship community. And we have a really diverse, vibrant scholarship community in Panama. So about 1,200 researchers come through Stride each year. About 900 of them are sort of young scholars. And I'm in charge of providing at least some stewardship for that community. So if you're interested in coming down to Panama, talk to me. I'll be here today as well as tomorrow. And part of my goal is to get more people doing science down in Panama. Okay, now our Earth contains roughly 6 million or so species of plants and animals. And I'm sure, as all of you guys can appreciate in this room today, all this diversity is packaged together in complex webs that interact with each other over local, over regional, and even over global scales. At one level, how all this marvelous variation in form and function on our planet is created is, I believe, the fundamental question in biology. It's certainly the question that underpins a lot of the biological research that we do across the Smithsonian. Now, I'm fortunate to work and live in Panama, so I work at the Smithsonian Tropical Research Institute. And it's hard to work in the tropics and be surrounded by this abundance of biodiversity and not to be innately drawn to this question. How does evolution generate biodiversity? And one way or another, this question really drives a lot of the research we do at Stride, and it's really the main focus of my research. So today, I want to celebrate one aspect of biological diversity, and that's the patterns on butterfly wings. What I want to do is sort of highlight some recent work, some published within the last couple of years, some will be published tomorrow, and some that's sort of half-baked, that's starting to get at a mechanistic understanding of how diversity is generated. Now, I'm an evolutionary biologist, so I'm coming at this as an evolutionary biologist, and I'm really interested in connecting genotype, which is the genetic composition of an organism, with phenotype, and how that organism looks or behaves, for example. Now, in the age of genomics, and I do a lot of high-end genomics like Paul, we've become remarkably adept at generating really fine-scale information on how individuals differ from one another, even species, at their genetic code. We have a much, much, much poor understanding of how these differences actually translate into the diversity we see in nature. Within this context, my attraction to butterfly wings is that they're simply wonderful systems for understanding how genetic changes generate phenotypic change, again, for linking genotype to phenotype. There are a number of reasons for this. Foremost, the patterns on butterfly wings are just simply large, simple morphologies. So they're roughly two-dimensional morphologies. They're created by these specialized cells called scale cells. And you can probably see them on this image of a wing there and there. And these scale cells are tiled across the surface of a butterfly wing in overlapping rows. So here's a magnification of a butterfly wing where you can clearly see these specialized scale cells. If you magnify even further, so now I'm looking at the center of one of these scale cells, you'll notice that these scale cells often have very elaborate microstructures, and they often contain a single pigment. Now, it's this combination of structural complexity and the pigment that allow these scale cells to both absorb and transmit light in wonderfully marvelous and fantastic ways. And so obviously, it's the aggregation of different scale cells across the surface of the wing that creates a pattern. And the wing patterns on butterfly wings are simply fantastically diverse. Recognize that the ability to color a wing using these specialized scale cells is a key evolutionary innovation of Lepidoptera, and nearly all 16,000 species of butterflies and 160,000 species of moth differ in some aspect of color and pattern. Now, these differences patterns are often under very strong natural sexual selection, and these forces often shape the variation that we see within and between species. And there's marvelous variation in nature. So for example, the wing patterns on butterflies have evolved a number of times to mimic eyes as a way to perhaps startle or deflect predators, or perfectly mimic the shape and pattern right down to the fungal decay spots of a dead leaf, or do exactly the opposite and mimic a living leaf. Thank you. Or the scale size can be greatly reduced so the butterfly becomes nearly transparent. In this way, it blends perfectly into its background. Of course, not all butterfly patterns are used for camouflage. A lot of them are bright and vivid colored, as in the case of this Heliconius butterfly, which is the group I do a lot of work on. So in addition to being sort of large, diverse morphologies, they're experimentally tractable in that pattern really maps directly onto a developing wing, and wing tissue is accessible all the way through development. So this sort of time series shows sort of wing pattern development. So the wing disc emerges from an undifferentiated group of cells in the larval stage, in the caterpillar stage, but by late caterpillar stage, it looks a lot like a wing. It starts off clear in the pupal stage, and then color and pattern get laid on later in development. Now here's a super cool video of this. This was taken by a student of Nipam Patel when he was at Berkeley. So you're looking down at a pupa. The outer cuticle case has been removed, so the wing starts out clear. This is the hind wing. And you see as it develops, it starts to turn sort of opaque, milky white as these scale cells are starting to mature and inflate. But really, color and pattern don't come on until the very, very last part of development, right about here, right before the butterfly closes, presumably in response to signals that were laid down much, much earlier in development. Now, the fact that everything's accessible to development really makes unraveling the networks of gene interactions that pattern the wing possible, and for understanding how they're modified to generate variation. Again, thinking about how you link genotype to phenotype. Now, amongst the different species of butterflies, butterflies within the genus Heliconius, I think, offer some of the most tractable systems for sort of really understanding how pattern variation is generated. Now, Heliconius is fascinating for lots of reasons other than color patterns. So, for example, this lineage of butterflies has evolved the ability to use pollen as a protein source. It's really the only butterfly lineage that does it. And associated with the evolution of this novelty, the group has evolved really huge brains. So these are smart butterflies. They learn where nectar resources are. They also have really long lifespans for butterflies, so they can live up to 8 months in the wild. They have very complex behaviors, and they have really rapid rates of speciation. However, it's hard to work on this group and not be completely fixated on the color patterns. So there are really hundreds of different color pattern types among the 47 or so different species that make up this genus. And the roots of research on Heliconius run really, really deep. They go all the way back to Henry Walter Bates and really the beginning of evolutionary theory and thought. Now, Bates got off the boat and into Amazonia when he was 23 years old. He spent 11 years in the Amazon collecting, and he had a particular fondness for butterflies, as well as a really keen eye. And in his vast collection, Bates was convinced that he'd actually captured the process of speciation by natural selection as it was happening. Now, and Darwin did as well, and Heliconius and other sort of butterflies in Brazil provided really the most visually stunning examples of speciation by natural selection at the time. It really helped cement evolutionary theory 150 years ago. Now, Bates was a beautiful writer. I mean, he was much better than Darwin. And I think he sums it up best when he writes, "It may be said, therefore, that on these expanded membranes nature writes, as on a tablet, the story of the modification of species." Now, the group continues to inform us about the process of speciation. And I encourage all of you guys to run out and grab last week's science issue that features work by Paul and others. And try to read it. You may get the math of it a bit complicated, but he's made a marvelous video of the process too. So go out and take a look at that. At their most basic level, the bright patterns like Heliconius serve as advertisements to potential butterflies, or potential predators that these butterflies taste bad. So as larvae, heliconias feed on a group of plants called passion vines that are toxic, and they either sequester or make cyanogenic compounds. This makes the adults, as well as the larvae, taste bad. And birds learn to associate a vivid pattern with a bad experience. And this translates into very, very strong natural selection in the wild. More recent work, a lot of it done at Stry, shows that these color patterns are also important in mate choice as well as species recognition. Now, this combination of natural and sexual selection plays out across the phylogeny to create a really interesting mosaic that I want to draw your attention to. So here's a cartoon phylogeny of the group. So this is some of the 47 or so different species and some of the 400 or so different color pattern types. And there's two dimensions to this mosaic. So as you move sort of vertically across this phylogeny, you see the same color patterns repeated over and over and over again. So distantly related species have converged on identical color patterns. At the same time, as you move horizontally across this phylogeny, so you're looking at closely related species or even populations of the same species, you see rapid color pattern divergence. So rapid color pattern divergence among closely related species overlaid on strong convergence between distantly related species. Now, this net result is a pattern convergence between distantly related species. If you go to a forest in the neotropics, so this is the forest, this is the long-winged butterfly community that you would find in, say, the Peruvian Amazon. The community sort of sorts into a handful of different mimetic types. We call these mimicry rings. Here are 3 of them. And notice that these mimicry rings can be quite diverse. So they're composed of not only heliconias, but also other more distantly related lineages of butterflies and even day-flying moths. Now, remember I told you that these butterflies are highly distasteful, and under Mullerian mimicry theory, which is really the first mathematical theory sort of in biology, under Mullerian mimicry theory, dangerous and distasteful organisms should converge on a common pattern. And so to a certain extent, Mullerian mimicry theory does explain the local pattern convergence you see between distantly related, distasteful butterflies. It doesn't at all explain a pattern like this. So here you have 2 species of Heliconius, Heliconius Arado on the left, Heliconius Mohamad on the right. So these are distantly related species. They diverged from one another early in the formation of the genus, about 12 million years ago. Yet within the last 3 to 5 million years, they've evolved a nearly identical patchwork of divergent color pattern races that change together as if, in the words of Henry Walter Bates, as if touched by an enchanter's wand. Again, he's a beautiful writer. Now, we've done a lot of work over the last 25 years to get at the genetic basis of the pattern variation we see within the group. And most of the variation that we see across the lineage is driven by variation around a handful of major color pattern loci. We often call these switch genes. These genes are on different chromosomes, so they're unlinked, and they affect pattern broadly. And by broadly, I mean both broadly across the radiation, so they work on a number of different Heliconia species, and I mean broadly across the surface of the wing, so they affect both pattern in the hind wing as well as the forewing. And they affect pattern, and they drive sort of the pattern convergence we see between distantly related species, as well as driving variation that we see within and between closely related species. Now, over the last 8 or so years, we've identified strong candidate genes for all of these major loci. So, for example, this gene called optics is what I call a red switch, so I divide them into color versus pattern switch. Last year, Marcus Comfort and colleagues made a strong case that Aristolus was actually the yellow switch that switches between yellow and white on the fore and the hind wing. A few years ago now, we made this case that this gene called Wnt was the forewing shutter locus, so it affects the distribution of melanin on the forewing and thereby affects the shape, size, the position of the forewing band. And finally, a few years ago, we made the case that this gene called Cortex was this sort of global melanin shutter. I don't know what else to call it. The nomenclature behind all these genes is really complicated, so I've tried to simplify it. Now, these genes join a really small handful of loci that are known to underlie morphological variation in nature. And their identification permits us to make a couple of broad conclusions about how pattern variation is achieved in Heliconius, as well as other butterflies and moths. And there are 4 take-home messages I'm going to leave you with today from this research. First is that evolution may be predictable, but it's still surprising. And I say it's predictable because the same genes are modulating pattern variation broadly across the genus. I say it's surprising because these are all very, very different types of molecules. And none of them, with one possible exception, had any prior link to being responsible for color and pattern in any organism that at least we were aware of when we were doing the research. Now, the second take-home message is that all the genes that we've identified as being involved in pattern variation in Heliconius are involved in pattern variation in other butterflies and even moths. So these genes act broadly across the Lepidoptera to drive the pattern variation that we see in nature. A perfect example of this was our discovery that this gene called Cortex was really a major target of wing pattern evolution in the Lepidoptera. Now, in Drosophila, Cortex is involved in cell cycle regulation, so how fast cells go through the cell cycle. In Heliconius, Cortex is this global melanin shedder. Again, I don't know what else to call it, but recognize that variation around Cortex is responsible for all the variation you see in these different sympatric color pattern morphs of Heliconius lumata. Variation around Cortex is also responsible for the increase in melanization in the British pepper moth. So how many remember their evolution textbooks, right? So British pepper moth is a classic example of evolution by natural selection. So the idea is that the black morph increased in frequency during the industrialization of Britain in the 19th century, only to decrease in frequency as Britain environment got cleaned up in the 20th century. Now the third take-home message is that there's really these are really highly conserved proteins, and there's really no amino acid differences between them, even between butterflies that look completely different. Moreover, they're used repeatedly through larval and embryonic development, and they're simply redeployed late on the wing to play a role in pattern formation. A great example of the diversity of roles of these color pattern genes is this gene called optics, which is the red switch again. So optics gets its name from studies of Drosophila that express this gene at a particular place and time in development. And that causes it's sufficient to generate a compound eye. Now optics is also expressed in the optic lobe of Heliconius, so it probably plays a similar role in eye development in Heliconius. But it also evolved a novel expression domain, so it's expressed in the wing about 48 hours after pupation, so again, the pupal wing is clear. And everywhere it's expressed in the wing becomes red in the adult wing. Which begs the question, how does such a small toolbox of really highly conserved genes generate the diversity that we see in nature? Now the answer to that question is that pattern variation is driven not by changes in protein structure, but by altering when and where these genes are expressed on a developing wing. So I want to dig into this last conclusion because it sort of underscores a general idea that's emerging from developmental biology in studies of morphological variation, and that is that a lot of the variation that we see in nature may be driven by changes in gene expression. And there are lots of different ways to change gene expression, right? And one of the ones that become particularly attractive is modifications in enhancers. Enhancers are a subset of regulatory elements. So they tend to be near genes. So I've shown you a cartoon here of a series of enhancers hooked up to a coding region here. And they're bound by proteins called transcription factors. And the idea is a transcription factor will sit on an enhancer and it will drive the particular gene expression in a pattern-specific way. They tend to be short and they tend to be arrayed in these sort of modular series, at least that's what we think. This is a photo of a textbook, a molecular biology textbook showing this gene even skipped, which sort of has the classic modular organization around enhancers. So you have a very strongly conserved coding region and a number of enhancers that flank it and drive the expression of this gene, again, in a pattern-specific way. Now, the reason why enhancers are thought to be such important evolutionary targets is that a gene can evolve new functionality by adding additional regulatory regions. So you can create novel patterns of expression without necessarily affecting existing patterns. And this, in theory, would incur less of a selective cost than if you altered the amino acid structure of a very conserved protein. Now, despite that compelling logic, and it is compelling, we actually know very little about how regulatory changes drive morphological variation generally, and nothing about what role they may play in an adaptive radiation like heliconias. So over the last several years, we've generated the genomic resources to start to identify the regions of the genome in heliconias that are actually driving the variation that we see in nature. And to do that, we simply leverage all the variation we have in nature. So we can go into the forest, we can collect butterflies that have these different patterns. This is all one species, Heliconias Arado. We can sequence their genome to really, really, really high coverage, and we can simply ask how genetic variation is associated with the phenotypic variation we see. In other words, we can use genomics to really explore phenotypic space. Now, we tend to focus on these two species groups, the Arado species group and the Mohamad species group. Again, these species groups diverged about 12 million years ago, yet within the last 3 to 5 million years, they've evolved a similar complement of identical wing pattern types. And by casting our results broadly, it provides really great context to explore the repeatability of evolution. I'm going to get into that a bit later. Not just at the level of the gene, but at the level of the regulatory switches around these genes, things that we think are driving the variation, as well as the networks that are ultimately responsible for patterning. And the data that we have comes from transition zones across Central and South America. These are zones of overlap between divergent color pattern races. There are dozens of these across the neotropics. Here's 4 or 5 of them for Heliconias Arado. And as Bates recognized, these transition zones can be really, really narrow. So you can go from a butterfly that looks like this, has a red 4-winged band, to a butterfly that looks like that, over about 10 kilometers. Moreover, and Bates also appreciated this, in the center of these transition zones, you get a complete breakdown of the phenotypic differences between the parental population. So and Bates interpreted this as speciation in action. What in fact has happened is lots of mating, lots of hybridization, lots of gene flow between these divergent color pattern races. And the practical advantage of that is that if you sample a butterfly on either side of these transitions, they're likely only to differ across their genome at regions of the genome that are responsible for the phenotypic differences that we see, right? Pretty simple. The data is more complex. So here's a really, really large data set presented graphically. So we're looking at about 100, more than 100 or so whole genome sequences from butterflies across the neotropics. So we sampled 7 different transition zones. So the wing patterns, and these are sort of Frankenstein butterflies. So on one side is the pattern that you find on one side of the transition zone, and the other side is the pattern on the other side of the transition zone. The x-axis represents the chromosomes of Heliconias, so there's 20 chromosomes plus the sex chromosome. And the y-axis is a measure of population differentiation. So the higher it is, the better, the more divergent the genomes are. Focus on the Panamanian hybrid zone here, which is a hybrid zone between a race, two populations that differ by the presence or absence of a yellow hindwing bar. Scan across that, really no differences anywhere with the exception of that one pull-up peak on chromosome 15. If you look across the entire data set, if you're still awake, you'll notice that there are a number of pull-up peaks across our 7 different transition zones, and all of those peaks fall directly on our color pattern region. So they're falling right on these color pattern switch genes that we identified. It's not the only place, so if you're really keen on these data, you'll probably notice that there's a region on chromosome 2 that's divergent between some comparisons amongst these different color pattern races. We think that's a large inversion that's circulating across Amazonia, and generally you see higher differentiation along the sex chromosome, which is consistent with population genetic theory. But around these color pattern switch genes are really the only places that you find nucleotide positions that are perfectly associated with wing pattern phenotype. So for this next slide, I'm looking at about a 200,000 base pair window around Wnt A. Remember, Wnt A is our 4-wing shutter locus. And you notice that these clusters of associated sites fall in non-coding areas around Wnt A. And again, these are, in this case, 5 different transition zones between divergent color pattern types. And we see exactly the same thing. In other words, clusters of associated sites in non-coding areas, in this case around optics, which is our red switch, as well as the other major color pattern genes. Now, our hypothesis is that these clusters contain the regulatory variation that alters the expression of these switch genes on a developing wing. And we further speculate that these regions are composed of a number of different modules or intervals, sort of similar to the architecture I showed you before around even skip. Each of those intervals is responsible for affecting the pattern in discrete areas of the wing, right? So what we want to do is try to identify smaller intervals that may contain the regulatory variation of these enhancers. Unfortunately, the patterns around any particular hybrid zone are really complicated. So there's groups of associated sites span tens of thousands of base pairs. However, by integrating across these different replicated and independent hybrid zones with a method of phylogenetic profiling, which we call TWIST, so a little bit of genomic magic, we can start to identify much narrower windows, so on the order of 200 to even 5,000 base pairs that are associated with specific aspects of color and pattern. So without diving too deeply into the details of the paper, as well as the method we published a couple of years ago, the idea is fairly simple. You look under these association peaks and you look for cases or places where you see a strong shift in the relationships amongst individuals from a tree based on geographic proximity, so that would be this tree here, which is found about across 99.98% of the genome, to a tree that groups individuals by color pattern phenotype. So for example, comparing the geographic tree to a tree that puts individuals together based on the placement of melanin across this part of the forewing, or across melanin on this part of the forewing, slightly higher, or slightly higher yet. So that's the logic, right? It makes a little bit of sense, doesn't it? And the result is we can start to really sort of, again, identify narrower intervals across these regions that are associated with specific aspects of color and pattern. And that's true across Wnt A, which is our forewing shutter locus, so we can identify a handful of regions that are associated, sort of color them here, that are associated with the place of the melanin in different places of the forewing. It's also true around optics. Again, you see really strong shifts in the relationships among individuals here, so that all individuals group together by the presence or absence of red on the hind wing. And similar, a shift here where individuals group together by the presence or absence of a red hind wing, a forewing band, and then sort of a more diffuse group of peaks that group individuals by the presence or absence of red in that part of the forewing. Now, across the radiation, we can identify really over a dozen of these intervals around our color pattern switch genes. And we believe that these intervals contain the different enhancers that, again, that drive the expression of these genes in a pattern-specific way. The net result of this regulatory architecture is that you can sort of move around a series of these enhancers around a handful of these sort of conserved genes and generate all the pattern variation that we see within Arado. So here's a simple model for thinking about this. So imagine a wing. This is about 48 hours after pupation, so it's clear. Now imagine a bunch of transcription factors that are expressed along the ring, and they have different expression domains. So you've got 4 different transcription factors. Now imagine you have hooked up to optics a number of enhancers, each of which that can read one of those transcription factors. The result of that is you can take a very conserved input, alter the enhancers that you have around your switch gene, in this case optics, and you can output very different looking butterflies. One that looks like that if you have those 2 enhancers, and one that looks like that if you have those 2 enhancers. Now, that model is almost certainly really naive. And our ability to identify these different enhancers is also really crude. However, you have to imagine that this provides really a powerful mechanism for generating novel variation. And we see that play out across the radiation. So for example, this population of Heliconia Arado almost certainly evolved from a hybridization between a red-banded and a red-rayed butterflies, that they all coexist in French Guiana. That hybridization event created a new regulatory module and outputted a different phenotype. We see exactly the same thing happen in Heliconius melpomene, again, the co-mimic. In this case, new wing patterns phenotypes are created by the movement of this functional variation across species boundaries. And across the phylogeny of closely related species in that group, there are at least 4 different cases of hybridization between species that allow these elements to cross species boundaries, caused pattern change, and probably promoted speciation in the group. Thus, again, the ready raw material for evolution exists, and new phenotypes can be recreated simply by reshuffling. Now, I like this quote because it summarizes what you need to do if you're interested in the evolution of morphological variation, and really highlights how far we've come over the last few years. So at this point, we've identified the different candidate loci genes and localized functional variation around them that we think drives most of the phenotypic variation that we see. We showed that these regions are composed of discrete intervals that are associated with different aspects of color and pattern. And finally, we demonstrate that reshuffling of this existing variation, as well as de novo creation of new regulatory variation, which I haven't talked about, allows for rapid morphological change. Now, this research was really hard, and it marks a really important step forward, but it's only the first step. And that's because, and this is important, genes don't make phenotypes. Genes mostly make proteins that interact with other proteins, or in some cases, other parts of the genome, to create these networks of interactions that interact with each other through development that ultimately creates pattern. Now, the overarching goal of this research has always been, and I've been doing it for a while, right, has always been to start to untangle those connections, to understand the links between those switch genes, those are the optics, the cortex, the Wnt, and the upstream genes that are signaling to them, as well as the downstream genes that are responsible for putting color and changing the microstructure of the scale cell. Now, to do that, we need to do 2 things. First, we need to obviously understand the regulatory variation around our color pattern switch genes in much finer detail. And secondly, we need to have the ability to actually modify the genome, both of which we started to do. So in the last 15 or so minutes, I'm going to sort of highlight some recent work that we've got. And this is will be published tomorrow, so it's just right off the press, in which we've developed CRISPR and used it to start to understand how these color pattern genes work. Now, I recognize that CRISPR is a technology that has lots of ethical issues associated with it, and I appreciate those. But as a biologist interested in variation, knowledge generated, it's simply a remarkable tool for understanding how things work. And the way, well, okay, for those of you who are sort of new to the technology, so it's pretty simple. So CRISPR uses a guide molecule, guide RNA, to recognize specific regions of the genome, and a cutting enzyme, in this case the Cas9 complex, that cuts the genome at that particular place. And then the cell's innate repair mechanism to basically modify the genome. So it's fairly simple and extraordinarily powerful, and it derives its power from its precision, its efficiency, and its relative ease of use. So in Heliconius, we raise lots of butterflies in Panama. We collect eggs right after they're laid. We inject our particular guides into an egg with the cutting enzyme. We let that egg hatch. We let the larvae grow. We let the butterfly close. It sounds really easy. It's really hard because a lot of these things die, but it works. And the results, I think, are fantastic. And we've done a lot of work on this gene called Wnt, which, again, I talked a little bit about. Now, Wnt is a member of a very, very conserved family of proteins that are involved in all sorts of functions throughout larval and embryonic development, including making a wing. So on butterfly wings, generally, Wnt is used to establish pattern. And in Heliconius, again, it's this four-wing shutter locus. So if you remember optics is expressed late in development. So in the pupal wing, Wnt is expressed really, really, really early in wing development. In the larval wing, so it's expressed about in the late 5th instar stage. So when the wing starts to look like a wing, and this is a blow up of this part of the wings in situ hybridization. So the dark areas represent where you find mRNA on the larval wing, and those areas tend to become melanized in the adult wing. So in this case, it prefigures melanin on the adult wing. So as a first step, what we've essentially done is knock out Wente across a dozen or more different Heliconia species, and really an equal number of different color pattern races or color pattern types within a species. And again, we're focusing on these two groups, the Arado group, which is shown in sort of light gray, and the Melpomene group, which is shown in the darker gray. So again, these two groups diverge from one another how many million years ago? 12, good. And within the last 5 million years, they've evolved a similar complement of color pattern types. Now, our experimental design is sort of cool because it includes 3 replicated cases in which these species from these independent lineages evolved identical wing pattern types. And these butterflies fly together in Panama, the co-mimics do. And they're really, really hard to tell apart. You can tell them apart, but the mimicry is really precise. It's very good. Now, the presence of these replicated cases of mimicry, I think, provide a really wonderful opportunity to determine if these identical wing pattern types are created using the same or different developmental pathways. So think of this as a type of replay experiment, that was made famous by Stephen Jay Gould in the early 80s. Now, Gould was a paleontologist. He was interested in the origin of animal body plans generally. He was also a really, really good evolutionary biologist. And he appreciated that evolution is a highly contingent process, where chance mutations and random genetic drift could really have a pretty profound effect on the outcome of evolution. So in his thought experiment, he reasoned that if you go back 500 million years ago to the middle of the Cambrian and basically replay the tape of life on Earth would look very, very different. Now, it's a nice thought experiment, but it's completely metaphysical. You can't do that experiment. However, convergent evolution across different branches of the tree of life does provide the opportunity to examine this tension between chance and determinism in evolution. In the case of comimetic butterflies, on one hand, because these comimetic pairs shared a common ancestry about 12 million years ago, which is really nothing in evolutionary time, you might imagine that evolution would be very, very predictable, and the same pathways would be used to generate identical wing pattern phenotypes. And if that's true, if we knock out a gene like Wente, we'd expect to see the same mutant phenotype, right? On the other hand, these two lineages were evolving separately for about 5 to 7 million years. And during that period of time when they were evolving separately, mutation and drift, as well as selection, could have caused the patterning networks responsible for patterning the wing to diverge. In that case, standard genetic variation would be different, and evolution might use different developmental pathways to drive the formation of identical wing pattern types. Thus, if we knock out a gene like Wente in the co-mimics, the pattern should be very different. So what do you think the answer is? Identical or the same? Identical? Sure. Good. Someone goes out on a limb. It's actually completely different. So in every species pairs that we looked at, the Wente knockout phenotype was different. And that was a surprise, a big surprise. So I'm going to show you 3 series of slides in which you have the wild type on the left-hand side and the Wente knockout mutant on the right-hand side. These are the 2 comimics, Arado and Melpomene in the case of the first slide. So in Arado, you knock out Wente, you get a large extension of red from the edge of the forming band all the way down to the base. In Melpomene, its co-mimic, the effect on pattern is much, much smaller. You don't see an extension of red, really, but you do see a sort of a shift from melanic to yellow scale cells there. It's even more dramatic in the mimicry pair Heliconia sapho and Heliconia sydno. Sapho is related to Arado. Sydno is related to Melpomene. Again, the knockout phenotypes are really, really, really different. So in the case of sapho, you knock out Wente, you get an extension of melanin all the way down to the loss of melanin all the way down to the base of the forewings, so an extension of white. Again, not much happening in sapho as a result of these Wente. When sydno as a result of these Wente knockouts, you lose melanin in this part of the wing, but not much change. You do get an effect in the forewing tips, so you lose melanin in exposed patches of white cells that you don't see in sapho. And interestingly, in sapho, you actually, on the hind wing, you get an extension of melanin, so you get a narrowing of this white hind wing fringe. So again, very, very different. And my favorite is the Wente knockout phenotypes between Huitzeni and Ficinus. So they're essentially mirror images of each other on the forewing anyway. So whereas you knock out Wente pectinus, which is closely related to Melpomene, you lose melanin in this part of the wing, so you get an extension of white that proximally or distally there. In pectinus, but not in Hewittsii. In Hewittsii, you lose melanin across this part of the wing, but not in pectinus. And in Hewittsii, you lose melanin to pretty much the base of the forewing. You lose a little bit in the base of the forewing of pectinus, but it's much more limited. So Wente is used to generate pattern variation in both species. It's just used very, very differently. And this knockout data sort of suggests the following scenario. And I'm going to lay this out over the next 3 or 4 slides, and you can tell me if you like this. And then I'll show you a movie. So imagine 12 million years ago, you're a common ancestor. We don't know what the wing looked like, so let's imagine the wing was, let's say it was clear. The two lineages diverged. They spent millions of years evolving separately during that time. The gene developmental networks responsible for developing a wing pattern diverged either through selection, drift, or mutation. The result of this is that when natural selection kicked in between 3 to 5 million years ago, it was kicking in on a different set of standing variation, and it drove the evolution of these identical wing patterns through different developmental networks. And the Wente knockout data really suggests that this has occurred across sort of 3 distinct developmental axes. So foremost, our knockout phenotypes really highlight a remarkable divergence in the regulatory architecture around Wente, right? Indeed, across the entire radiation, Wente is expressed and affects pattern variation broadly, essentially from the tip of the wing to the base of the wing. This is true for both the fore and the hind wing. So here's a slide, a little bit complicated, but this is Wente knockout phenotypes of 9 different Heliconia species. These are Frankenstein butterflies again. So on the left-hand side, you have the wild type phenotype, and on the right-hand side, you have the Wente knockout. This cartoon sort of represents the difference in patterns. So it represents the functional effect of knocking out Wente. And if you look across the entire genus, so across about 12 million years, you see that when you scan across these different knockouts, you see that Wente is really from the base all the way to the tip of the forewing, as well as from the base to the tip of the hind wing as well. And that's true across the genus. It's also true across the Arado species group, so across about 5 million years ago. And it's arguably true across the Melpomene species group, which represent about 3 million years of evolution. So it's really affecting pattern variation extraordinarily broadly. So in addition to affecting pattern variation broadly, Wente also affects the ultimate color fate of a scale. So you can think about this as basically determining scale fate. And on a Heliconia wing, scale cells are fated to be one of 3 different types, either black, red, or yellow. So white is the absence of pigment. And Wente, depending on its genetic background, alters the ultimate scale cell type in every possible direction. And that observation reveals, I think, a second axis of divergence between the co-mimics. And that's how Wente signals are actually interpreted by downstream genes. And finally, the Wente knockouts highlight a third axis, and that's the presence of other patterning loci that, similar to Wente, affect pattern variation on a Heliconia wing, but they do it differently. And a perfect example of this is looking at the genetic basis of this patch here, which is when you knock out Wente, it's clearly under different genetic control in pectinus than it is in Hewittsii. Again, these 3 different levels. Thus, even in a really recent radiation, only 12 million years, under a very, very deterministic scenario of evolution, mimicry, where natural selection is really pushing everything to the same fitness optimum, there's surprising unpredictability in the paths taken by these different species to achieve convergence. So if you hate that figure, and reviewers did, I didn't like it because I drew it. We made a movie, so this paper should come out tomorrow, I think, in Current Biology. So imagine that you have Arado and Melpomene. This is appropriate for this group because you have all these mountains around here. I'm not used to seeing them. But 12 million years ago, they start out the same place. But they take different paths, right? So they sort of diverge. They start moving apart. Sometime within the last 3 or so million years, natural selection pushes these species both to the same fitness peak, but because they've moved they're starting at the base of the mountain from a different position, they go up that peak using different pathways. Now, on a more general level, our CRISPR work, I think, really underscores just how deeply embedded Wente is in wing patterning networks, how deeply embedded this gene is in the wing patterning networks of heliconias. And I'm going to leave you one final analogy, and I'll present the last little bit of data. So imagine a paint-by-number butterfly, like the one that you used to color, or maybe you color with your kids, or maybe you still color, I don't know. So in this case, sort of Wente is affecting pattern variation by not only moving the numbers around the cells, around the butterfly, but also changing the corresponding color that is connected to each number. And it illustrates, I think, in a marvelous way, this is a quote from one of my collaborators, how the repeated tinkering of a developmental gene can foster really extraordinary evolutionary change. So I can imagine Bates saying that. And that is actually in a paper, so that's cool. So in their last few minutes, I want to introduce some really recent research efforts. So this is unpublished stuff, right? So this is stuff that we're starting to do now, and we hope to get out in the next year, that I think really holds exciting promise for really understanding the regulatory landscape around these switch genes like Wente, and how it's modified to create the variation that we see. And this research is really coupling the genome-wide association data that I showed you before with a new high-throughput method of characterizing the regulatory landscape in open chromatin, called a TAC-seq. Now, similar to most of these sort of high-throughput methods, the way they work is that sequencing reads sort of pile up on regions of the genome in open chromatin that are likely to be regulatory regions. So this slide shows data from a number of these different technologies for surveying that. And the only thing to really recognize, this is around human chromosome number 20, I believe, maybe it's 19, is that where you see these peaks are putative regulatory regions, right? Either promoters or enhancers. So the idea is things are bound to them. Now, what the really fantastic thing about a TAC-seq is that these peaks are really repeatable. So here's the TAC-seq profile across that region relative to the DNA-seq profile. So DNA-seq is just another technology for doing this. It tends to be the gold standard for figuring out, for profiling the epigenetic landscape. And the peaks are identical between a TAC-seq and DNA-seq. However, the DNA-seqs are generated using about 2 to 3 orders of magnitude less cells, which is really important if you're working on a butterfly wing. So we've, over the last year, generated really lots of data. I'm going to show you data from 2 individuals. And this is a TAC-seq profile across the part of the genome that contains Wnt in 2 different species, Arado and Mohamad. And in this case, 3 different types of tissue: the forewing, the hindwing, and the head. So our head is our sort of control, right? And now we've done, at this point, hundreds of these, and it's very repeatable and robust. So we get the same pattern between individual species that look the same, and different patterns between individual species that look different. And there's a couple things I want to draw your attention to. One is that it's nice because it generates what we'd expect. So we can identify the putative promoter region here. You see that both in Arado and Mohamad. These red lines sort of are putting together homologous regions because the genomes are slightly different between the 2 species. So it's nice. It's generating what we'd expect to do. And if you scan across that, you see a few peaks here that you get really excited by. So there's this peak here that falls right in the middle of that genome-wide association peak. It's associated with the presence of melanin across this part of the forewing, right? And it's nice. So it's in the forewing and the hindwing, but it's not in the head. So it appears to be a species-specific regulatory region or a tissue-specific regulatory region. We do not see that in Heliconius melpomene, which we wouldn't expect to see in Heliconius melpomene because we don't see a knockout phenotype. But that generates a prediction, right? That prediction is if we go in and use CRISPR and cut out that peak, we should see a change across this part of the wing in the patterns of melanization. So just to remind you what the wild type looks like versus the Wnt mutant phenotype. Again, the Wnt mutant affects pattern all the way to the base of the forewing. It also has a large effect on the size of the hind wing bar. And again, we would predict if we knocked out that peak based on its association and where we find it, we'd see area of melanization in this part would be being affected, which is exactly what we see, right? So we're starting to actually generate testable predictions and using CRISPR to test them. So at this point, I hope I've given you some understanding of why I consider butterfly wing patterns to be really extraordinary systems for linking genotype to phenotype. I've given you some appreciation for maybe how far we've come over the past few years and our efforts to gain a mechanistic understanding of how pattern variations are generated on the butterfly wing. There's obviously lots of work to do, but I think the research directions are clearly laid out, so I'm very excited. And finally, I hope that I've started to convince you that, at least to some extent, the prediction that Henry Walter Bates made 150 years ago, that at some point the study of butterflies would become one of the most valued and important branches of biological science, is starting to come true. And with that, I need to thank all of my collaborators. I've had a wonderful scientific career, and I've been fortunate to interact with lots of scientists in all parts of their career, from interns who are learning how to do science to graduate students to postdocs, as well as having just a great group of long-term collaborators. And this is really a collaborative effort, and it's been fun to see these different threads come together over the last 10 or so years. So with that, I think I'll take any questions. Thank you.
All right, suppose we have to leave, go ahead, but we'll take a couple questions.
Thank you for showing the variation within Heliconius itself. But Heliconius is also mimicked by a variety of other families and succession. You alluded to that at the beginning. Have we got some of the same tests in those other families? See how they might have generated with those speciations?
Yeah, so we're starting to do that now. So there's lots of ethonions and that we can rear. So the nice thing about living in Panama is we're in a forest, and the butterflies do mostly well other than that.
Where do you live?
So I live in Gamboa.
Okay, good.
So I live right next to the new research lab and right on the border of the national park. So yes, we're starting to do that now. That's the next step to sort of see, how we know that Wente is used across nymphalids. So we published a paper a couple of years ago in PNAS, which was really the first paper, which did exactly this, but looked not just on Heliconius, but looked at nymphalids generally, and affects pattern in really remarkable ways across nymphalids. So for example, in monarchs, it causes, it also affects pattern. It causes changes in the distribution of melanin as well.
Is there any indication that those changes are being transferred in a reticulate fashion, that we have horizontal gene transfer at the level of family?
The level of family, no. But
hybridization so far.
Yeah, certainly within the Arado group and the Melpomene group. So the great thing about Paul's paper is that we knew there was lots of hybridization going on within that one species group, the Melpomene group, because they're closely related. You find hybrids in nature. There's evidence for hybrid species. We've known that for now about a dozen years, and we had a feeling that was happening for 25 years. So we showed that. What Paul and collaborators did, and actually I was one of the collaborators, I didn't have anything to do with the paper though, really, showed that within the other group, within Arado, there was also strong evidence for hybridization and transfer of genetic material across species boundaries, and it really played out generally across the radiation, and important for adaptive change as well. So, but between butterfly, different genera, nothing I'm aware of.
Take another question. Yeah.
So yeah, that was beautiful work. I really liked it. Amazing. But the cis-regulatory element focus makes a lot of sense. There's a lot more flexibility there, right? That's the standard argument.
Yeah.
But I did notice that several, at least as far as I interpreted the graph, several GWAS peaks landed right in the gene itself, the Wente gene itself.
Yeah, I mean, it lands around it, but those associated sites don't cause a change in amino acid. So,
Okay.
and those sites across any transition zone, it's ugly. But yeah, there's no amino acid differences between these things. So it's just that when you look at a GWAS peak across the transition zone, we just don't have the recombinations to narrow it across any single one. And we did this with just a few individuals on the order of 10 individuals. I mean, we can go back into nature and sample hundreds of individuals, and I think we would get a much better understanding of where these
Okay, yeah, so you really do think it is, at least in this system, it's all cis-regulatory?
Well, I mean, I think there's some trans effects too. I just can't get my head around those. Those are hard to me to think about. But at least at the level of switch genes, I think it's cis.
All right, thanks, Owen.